All right, so in this video, we're going to be wrapping up chapter 15. The first part was on sex-linked traits. The rest of this is just going to be about um, other alternative ways that we study inheritance. So um, it's pretty rare that you would get a genetics practice problem on something like this. A lot of this is just going to be about, um, I would assume, some type of analysis of data. So that's what this one's going to be about. So the first thing we're going to talk about are linked genes. And basically what this reminds you of is a couple of things that you need to remember is that chromosomes contain hundreds of thousands or even thousands of a particular gene, right? So this is just showing you that a chromosome has two genes, the A gene and the B gene. So genes on the same chromosome are usually inherited together. We're going to refer to those as linked genes. And in terms of crossing over in meiosis, you might want to go back and revisit meiosis and crossing over and what that is in chapter 13. But usually when they're inherited, if they're closer together on the chromosome, the more likely they are to be inherited together. So if they're not inherited together, there's a couple of things. That means crossing over, excuse me, if they're not inherited together, if crossing over takes place, right? So depending on if those genes actually do cross over, you get some new recombinant types. So the way that you should think about this, and here's a really common example, is red hair and freckles. Oftentimes those are inherited together for um, really a location on the chromosome is going to be the reason why they're inherited together. So notice that the genes for A and B, let's say the A gene represents having red hair, the B gene represents having freckles. Keep that in mind that even if you do crossing over in meiosis, oftentimes that these two are going to be inherited together. So the only way to split up those genes is if crossing over occurs between them. So if no crossing over takes place, and we're going to have non-recombinance. Now, even if you cross over and the genes are super, super, super close together, oftentimes they're inherited together. So let's kind of show you what this looks like in terms of a practice problem. So let's go back to fruit flies. I have VG plus is wild type for wing, which is normal. VG is vestigial wing, which is kind of a shorter version of the wing itself. B plus, the plus means it's wild type, and that's going to be dominant. And lowercase or just a little b represents black body, and that's going to be recessive. Okay, so if a fly with the genotypes were crossed, here's kind of what we were looking at, and this will kind of help you understand what you should be able to catch in the problem. So let's say this is the genotype, B plus B, so heterozygous for black body and heterozygous for wild type. So we have a wild type. This one would be gray. This was the parental type, right? Gray and normal. And we crossed it with black vestigial. Okay? So black and vestigial. So let's kind of figure out what we did here, or what we're going to do here. So notice that this fly is going to be dominant, but it's heterozygous for those dominant traits. And then the black vestigial is homozygous recessive for both. So what's the probability of these possible scenarios? Well, to do that, let's figure out our prediction here, and I kind of did it over here. Just to remind you, we're doing the practice problem here where we're actually doing the probability method, and that's how we'll solve these. So if I just cross the B's over here, I crossed heterozygous for, this one is talking about B plus B. This is wild type, so this would essentially be wild, which would be gray body. And you crossed it with black. So these are the results. We have 1/2 would be wild. We also have 1/2 would be black body. I did the same thing over here with vestigial, so this would be wild type. So again, wild wing size, and then this would essentially be your vestigial. And again, I did those monohybrids, and then it just becomes a simple probability method, okay? Now, the reason why I wanted to do this one first is because notice that this parent was heterozygous for both traits, and this was homozygous recessive. So what's the probability of being gray normal? So let's look at this. So we have to look the probability of getting gray normal. So that would be B plus B, and normal wing would be VG plus VG. That's the only possible genotype. I couldn't get B plus B plus because according to my Punnett square notice, when I did it, there's no way, according to the parent genotypes, I could get B plus B plus. So this is really the only genotype that I could get. So what's the probability of being gray at normal wing? Well, then I just look at these ratios I figured out. B plus B was 1/2, so it'd be 1/2 times VG plus VG, VG plus VG, so that's wild wing, times 1/2. So I got the answer of 1/4. So I have a 1 in 4 chance, or 25% of my population should be that phenotype. What about being black and normal wing? So for this one, they would have to be this genotype, a little b, in VG plus VG. I'm going to look back over here. They're all 1/2. So black would be 1/2 times VG plus VG, which is also 1/2. So it would also be 1/4. Now, hopefully you're able to pick up on this and you can look at this on your own, or solve it. But basically, they're all going to be 25% or 1/4, right? And that's my prediction. Okay, so that's what we did here. I have my ratios, and that's how I solved those. So there's a reason why I wanted to do this, is because let's say if you got a problem and you cross the fly like this, and this is one thing that we need to consider here. If these were my two, right? If I cross this fly, this is the exact same setup as that previous problem. I crossed a black with vestigial wings. That was this fly. I crossed a wild type, so that means gray color, as well as normal wing. When I crossed them, just like we did over here, we expected to get a 1-to-1 to 1-to-1 ratio, right? A fourth to a fourth to a fourth to a fourth is a nice 1-to-1 to 1 ratio, okay? Now, this is something that we need to consider. So basically, if you had a population size and you solve this and you took these two flies and you're expecting to see this nice 1-to-1 to 1-to-1 ratio, that means that the genes for wild type, or the genes for, the genes for color, colored body, whether it's black or gray, and the genes for wing type, whether it's vestigial or normal, that's what we expected to see, that they're going to be on different chromosomes. But look at what actually happens is that you get a really high number here. That was what you expected according to your Punnett square. But if you actually observed a really high percentage looking just like the parents, right? Because these two look just like the parents, wild type, and then black and vestigial. You get a really low number of a combination where you get gray vestigial, right? Because that didn't look like either of the parent, it was a combination, and then black and normal wing. None of the parents look like that. So these are going to be recombinant, and they're really, really low. And when you see that happen, if you look at that, that means that typically that those genes then must be linked on the same chromosome, that they're often inherited together, that even when you cross them over in meiosis, they're inherited together. So that's kind of the type of problem that you would have to be able to identify, that according to your Punnett square, you expected to see a 1-to-1 to 1-to-1 ratio, but you get a really low percentage of recombinants. Now, if you don't know what recombinants are, that means the offspring's phenotype is different than the parents. That's what recombinants are. And if you get a low percentage, so if percentage is low, that means that the genes are going to be linked. So that's what I wanted to point out on this one. And again, when we say that they're linked together on the same chromosome. So rather than talking about red hair and freckles on this example, if we're talking about the fly, this could be the vestigial and color. Okay, those genes are located on that same chromosome. So that's really what we look at, what we think of if they're linked or not. Oftentimes we have to look at data of the offspring, and that'll help determine it. Okay. And this is just kind of clarifying what's going on here, is that crossing over takes place in prophase 1, and it's within non-sister chromatid. So that means it's the homologous pair that's crossing over. So let's figure out linkage mapping, and they're actually pretty easy. And this just kind of goes to show you a couple of things here. So if you look over here, this is the original chromosomes, right? These are homologs. Remember that DNA has to replicate, right? And right here, I still have some crossing over. Now, the one thing I want you to take a minute and notice is that look at the genes. We have the A gene, the B gene, and the C gene. C is really far down that chromosome, right at the tip of that chromosome. So it makes sense that those are going to get crossed over. But notice A and B are typically going to be close together. All three of these genes, A, B, and C, are considered linked. But one thing that we got to look at is how to figure out a map unit. And we look at your recombinant frequency. So basically, you need to think of that as the percentage of crossing over. So that's what we need to think about. So the more likely they're crossed over, the further apart that these genes are going to be. So A and B, you're not going to get a really high recombinant frequency due to crossing over because they're so close together on that chromosome. So when you think about it, it makes sense. If the C's are really, really far at the end of the chromosome, you're going to get a high percentage of crossing over. So let's show you how to do a practice problem. So all of these, what is the correct order of the genes? What are the map units here? These percentages are basically showing you the percentage of crossing over. And really all we're trying to do here is figure out what's the order of the gene, and it's actually pretty easy. So you're going to draw a line, and you always want to start off with the highest percentage because those are the genes that are going to be the furthest apart. So basically what we're doing with this is we're trying to figure out, like we had here, how do they know it's A, B, and C? And this is what geneticists do when they look at data like this. We're just figuring out what's the order of the genes. So what I like to do is I'm going to put the B gene and the D gene over here, and I'm going to keep these as far apart as possible. And I'm going to record this as being 23 map units apart. So I'm going to put this right here as 23. Okay. Percentage of crossing over in map units, they're going to be the same number. So that's our first one. So let's try to solve for the next one. Well, I'm going to look right here. B is really, really close to the C gene because they only cross over about 4% of the time. That's a really low percentage. So that tells me that if I put C right here, they need to be 4 map units apart. So I'm going to put this right here. And you don't have to do this. I just like to check my work. B and C are 4 map units apart. So let's kind of figure this one out. Well, if this is 23 map units apart and I have 4 map units accounted for here, do I have any data about C and D? I don't. Okay, I do not. But do I have any data about A and D? Yes, I do. So that's 4%. Now, let's figure this one out now. I'll put A right here. And it tells me that A and D are really close to each other because they're only 4% apart, or 4 map units apart. So I'm going to put that here. So here's my genes. I'm going to double check this a couple of different ways. So let's figure this out. What else? What other pieces of data do we have? Well, it says A and B are 19 apart. So I'm going to go and double check this. So if these are 19 map units apart, we know that 4 map units apart are going to be A and D. So what's 4 plus 19? It's going to be 23. And that is a correct order. So what is the correct order of the genes? It would be B, C, A, D. Or you could have it written in the reverse that it goes D, A, C, B. Okay. Either of those would be correct. It's just how you started out with your problem. But that's how you would actually map a gene out. Not too terrible. Start with the highest percentage and then go from there. It's not terrible. Look at the data. Make sure it all adds up and makes sense. Now this last one is really just talking about alternative types of sex chromosomes. So the one that you're going to be most familiar with is going to be the XX and the XY, where the males are going to be more likely to express a trait. But there's other types of species that have different sex chromosomes. So we have the X and the zero system. So some insects, grasshoppers, and cockroaches, males don't even have a Y chromosome. They just have one X chromosome. So rather than being XY, they're just X. We have the ZW system, birds, fish, and some insects. This was the FRQ a couple of years ago. You didn't have to memorize it. It kind of gave you a scenario where it kind of clarifies what's going on. But in this case, it's interesting that The males are the ones that are homozygous, and the females are the ones that are actually going to be more likely to express a sex-linked trait. So other than that, we also have the haplodiploid system, bees and ants. There's essentially no sex chromosomes, where the bees, like a lot of the male worker bees, are actually haploid. The ones that are actually diploid are going to be like the queen bee. So kind of interesting. There's all kinds of different inheritance patterns. So this is just further talking about the same thing, different disorders, color blindness, muscular dystrophy in humans. But I do want to mention non-disjunction here. And let me just kind of clarify why this matters. This is typically going to be an error. So an error in meiosis 1 or 2. Okay, so basically what's happening here is that the chromosomes get separated incorrectly. So basically what's going on here is that if you had these two chromosomes lined up, if this was going to be meiosis 1, right? If you look here on the left side, they separate normally, okay, meiosis 1. But if you look at this line on the right, notice that when they separated, you get this one that is actually faulty, okay? So you normally should get some chromosomes here, but they don't. They all got divided, and then you were resulting in some faulty cells, okay? And oftentimes you get what's called polyploidy, multiple sets of chromosomes. Really common in plants, like strawberries that you eat are octoploid. That means that they've done meiosis incorrectly, but it didn't hurt their ability to survive and reproduce. But basically, it's just when your chromosomes separate incorrectly, and this can happen incorrectly on. If you look at the one on the left, this is showing you that meiosis 1 happened correctly, but in meiosis 2, this happened fine, but it looks like at this cell, when it did this separating of your sister chromatids, they didn't separate correctly. A really common example of that, I shouldn't say really common, but one example of this would be people or anybody that has Down syndrome would be just a small result of non-disjunction in meiosis. They got that extra chromosome. And that was just due to an error in meiosis that they got that one extra chromosome from how these chromosomes were separated in meiosis. So that's what non-disjunction is. You get an error. Depending on the chromosome that's inherited, sometimes it can be a complete viable offspring. Sometimes that it doesn't become a viable offspring. It just depends on how many chromosomes they had extra. So that's non-disjunction. And like I said, it can happen in meiosis 1 or meiosis 2. Chromosome alteration. This is talking about specific genes. Entire genes can get deleted. Some genes can get duplicated. Sometimes we can actually invert them. So rather than being BCD, we can actually be DCB. And then we get reciprocal translocation. That's where a gene that's on one chromosome actually gets moved over to another. Now, in terms of that, this just kind of reminds you of, kind of what I was talking about on the non-disjunction. So from non-disjunction, Down syndrome, trisomy 21, you get an extra one on that 21st one. This is showing you Turner syndrome. So this would actually result in somebody that's sterile. They only have one X chromosome. Females that can actually have trisomy X, where they actually have an extra X chromosome. And Klinefelter syndrome would essentially result in a male, but actually sterile because they have an extra X chromosome. They have an odd number of chromosomes. So that's where they do result in some sterile gametes. So other than that, I think that's kind of the last bit of key information that you guys need for chapter 15. I hope that was somewhat helpful.